Scientists have created a synthetic life form with just 57 genetic instructions. Here's what you'll learn when you read this story. While most organisms on Earth share a basic genetic code, it's not perfect. Scientists wanted to fix these flaws by building an artificial genome that replaced E. coli's original one using 4 genetic materials. This innovation could lead to superorganisms that resist viruses, among other benefits. The genetic code for nearly all life forms is remarkably universal. https://www.popularmechanics.com/science/environment/one-single-ancestor/life-Luca-discovery. It uses 64 codons, short sequences of nucleotides, that encode amino acids, which build proteins and form the backbone of DNA and RNA. Why would anyone mess with something that's been unchanged for billions of years? Despite nature's reasons for preserving some things, biologist Jason Chin from Oxford University decided to experiment. Some codons carry extra instructions for building proteins or signal when to stop protein production. Chin wanted to see if life could function with less. He already proved that Sin61, a bacterial genome with 3 fewer codons than before, worked. Now he's taken it further with Sin57, a genome of just 57 codons. Reducing the number of codons can help some organisms fight viruses. When synthetic genetic codes are transferred to cells, they block viruses by altering their protein-making processes. This could also boost protein efficiency and help create long chains of amino acids, over 500 in length. Protein polymers, engineered repetitive polypeptides, can mimic natural proteins but are also customizable to solve molecular problems. Chin and his team, in a study published in Science, https://www.science.org, DOI 10.1126, Science ADDI 4368, said synthetic genomes might differ from naturally evolved ones. Genome synthesis offers a way to design sequences and functions that don't exist in current life. Earlier research removed stop codons, signals that end protein production, from E. coli's genome, often through mutagenesis or gene editing. But mutagenesis has downsides, excess mutations, and difficulty balancing sense codons that make amino acids versus stop codons. Sin61 showed organisms could survive with 61 codons, but was that the limit? In creating Sin57, Chin altered over 100,000 codons, some needing tolerance across entire genomes, others just a single replacement. He assembled the DNA into an artificial chromosome, introduced it into E. coli, and tested its growth. Some strains of E. coli were cultured before the final version emerged. When cloned, future work will explore deeper edits to genetic codes, enhanced virus resistance, and new proteins/polymers. In future work, we'll build on this deeply recoded strain to explore deeper edited genetic codes, Chin said. This synthetic life replaced its genome rather than being created from scratch. Could it reveal how life and its genes originated on Earth? Elizabeth Rain, a writer who contributes to Popular Mechanics, Ars Technica, and other outlets, lives in New York City with her parrot, Lestat. When not writing, she draws, plays piano, or shapeshifts.